All right, chapter 26, the last chapter of the book, Reproduction and Development. So this will be included on your final. There's only 4 chapters on your final. It'd be the chapter on renal function, the chapter on balance between water balance and pH balance, and then the chapter on digestion, and then this chapter on reproduction. But the first thing we're going to talk about is what determines the sex of the fetus, and it boils down to their chromosomes, whether they have a Y chromosome or not. But the sex organs are 3 sets of structures. First, we have gonads, and the gonads are the primary sex organs, and this is because they make the gametes. I forgot to mention earlier, but you might want to go ahead and review the anatomy of the male reproductive system and the female reproductive system. So as I go through this, if I happen to say something like seminiferous tubule or ovarian cortex or cervix, you're going to know what I'm talking about. All right, so gonads. Go back over that real quick. They are the primary sex organs for males and females. The reason why they're called the primary sex organs is because that they are the organs that make the sex cells, which are going to be the gametes. The difference between a gamete cell and a somatic cell has 46 chromosomes. Those are your body cells. Gametes only have 23 chromosomes, so the number of chromosomes is knocked into half. And that's because whenever the gametes meet for fertilization, that you'll create a zygote or fertilized egg with 46 chromosomes instead of 92. Anyway, so the gonads, they are producing the gametes, and they also produce the sex hormones like testosterone and estrogen and progesterone. So the male gonads are the testes or the testicles, and they're the ones that produce the sperm. Sperm are the gametes. Female gonads are ovaries, and they're the ones that produce the egg cells known as ova. Ova is an egg cell or ovum. And then they have internal genitalia. Those are going to be accessory glands to the reproductive system and ducts. For the male reproductive system, these glands are producing secretions that eventually become semen. And then in the female reproductive system, these glands are producing lubricating secretions. And then the only thing left are going to be external genitalia. Most of the external genitalia is found outside of the pelvis, but there are some external genitalia that's found on the inside as well. This next part's a pretty easy read, so let's just hit the highlights. Normal nucleated cells, the somatic cells, they have 46 chromosomes. And they call our chromosomes diploid, which means we have 2 sets of them. The DI in front of that means 2. So 2 sets of chromosomes, so we have 23 pairs. 22 pairs of those chromosomes are called autosomes. And those are the parts of your genes that are dictating how tall you're going to be, what color your skin's going to be, what color your eyes are going to be, whether you have attached lobes or unattached lobes, so on and so forth. Those are the autosomes. You have 1 pair of chromosomes, which are your sex chromosomes, and they're going to be some sort of variation of X and Y. Females have 2 X chromosomes, so they are designated XX. Males have a Y chromosome, and they're designated as XY chromosomes. So in the formation of gametes, the formation of the sperm cells and the egg cell, they're only going to have 22 autosomes, which are going to be 11 pairs, and they're going to have 1 sex chromosome. A Y chromosome is the chromosome that's going to determine whether it's going to be a male or female fetus. In the presence of a Y chromosome, the embryonic sex organs will develop into male sex organs. In the absence of the Y chromosome, then those sex organs will develop into female sex organs. And since it is the Y chromosome that determines whether the fetus will be male or female, then the sperm cell is the cell that's going to determine the sex of the zygote. The zygote is the fertilized egg. All eggs are going to have an X chromosome, and that is because as the gametes are formed, the female chromosomes do not have the Y chromosome, so every egg is going to have an X chromosome. In males, with the sperm cells, half of them, 50% of them, will have the X chromosome, and 50% of them, the other half, will have the Y chromosome. So if the sperm cell is the cell that, the sperm cell with the Y chromosome, if that is the sperm cell that fertilizes the one egg, then the fetus develops into a male. And that's what we're showing here in this picture. We have the mother and we have the father. This is the designation for the mother. This is the symbol. And then here's the symbol for male for the father. All of the egg cells are going to have an X chromosome. Half of the sperm cells will have an X chromosome. Half of the sperm cells will have a Y chromosome. Now if the X from the egg unites with the Y from the sperm, then it forms a male fetus. If the X on the egg joins with the X chromosome of a sperm cell, then it forms a female fetus. For the first 7 weeks after pregnancy, the sex organs are called bipotential gonads, and that is because they can either become an ovary or a testes, depending on whether it has the Y chromosome or not. And then there's a whole series of ducts that are going to be similar, and they're bipotential ducts. We have Wolffian ducts and we have Mullerian ducts. One of them will become the male reproductive system, the other will become the female reproductive system, again depending on whether it's going to be a male or female zygote, or fetus I should say. And then also what we have in common for males and female fetuses is that they all have a genital tubercle, the urethral folds, the urethral groove, and then swellings called labial scrotal swellings. Now again, what's going to determine whether it's going to be male or female will be the Y chromosome. So that is why the males and the females in humans have homologous organs, if you remember that from anatomy. Homologous organs means that they had a similar origin, but they are different, or they diverged into different organs. For example, the scrotum in the male reproductive system is homologous to the female labia majora. Since it is a male, if it is a male fetus, then the labioscrotal swelling develops into the scrotum. If there is no Y chromosome, then the labioscrotal swelling develops into the labia majora. On the Y chromosome, there is a gene called the SRY gene that stands for sex determining region on the Y chromosome. That is going to make the Wolffian ducts develop into the epididymis, the vas deferens, and also the seminal vesicle. Now, if you remember what they are, the epididymis was the structure that's on the posterior surface of the testes, and it's where sperm cells mature. The vas deferens, also known as the ductus deferens, that is the duct that it leads from the epididymis into the ejaculatory duct. And then the seminal vesicle, that was a gland that was located on the posterior surface of the bladder, and it produces seminal fluid. If the fetus has a Y chromosome with the SRY gene on the Y chromosome, so that means the Wolffian ducts will develop into the male reproductive system, and then the Mullerian ducts are going to degenerate. If the Y chromosome is present with the SRY gene, it produces a hormone known as AMH. AMH is known as anti-Mullerian hormone because the Mullerian ducts will develop into parts of the female reproductive system. So if they have the Y chromosome, they're going to make the Wolffian ducts proliferate and develop into the male reproductive system, and it's going to make the Mullerian ducts, which would be part of the female reproductive system, to degenerate. And then the testes will develop later. You'll note that testosterone is not required in order for the testes to develop, and that is important because the female, the mother, does not produce testosterone. So the testosterone will have to be made later, and that's after the testes have been developed. And then the testes are going then to descend into the scrotum, and that is after the presence of testosterone, which isn't going to be produced by the functioning testes until right before birth. There are cells located inside of the scrotum called Sertoli cells, also known as sustenacular cells. These Sertoli cells that are eventually going to become the sustenacular cells, they are the cells that secrete the AMH, the anti-Mullerian hormone. And then also inside of the scrotum is going to be the interstitial cells. The interstitial cells are the cells found inside of the seminiferous tubules, and they secrete the male sex hormones, the androgens. They secrete testosterone and something else called DHT, which is dihydrotestosterone, which will eventually become testosterone once it's been changed. So if there is no Y chromosome, which means there is no SRY gene, then the embryo automatically develops into a female embryo. That means the Mullerian ducts will develop, and they develop the upper parts of the vagina, parts of the uterus, and also the fallopian tubes. That is because there is no AMH, which would have been developed from the Sertoli cells if it was male. The Wolffian ducts are going to degenerate. That's because there's no testosterone. And then the external genitalia will develop into the female reproductive system because there's no DHT. So it's basically what a fancy way of saying what I said earlier. If there's a Y chromosome, it changes the fetus into a male. If there is no Y chromosome, everything stays the same, and the embryo develops into a female. So for the visual people, we we have the bipotential gonads. We have the Müllerian ducts, and we have the Wolffian ducts, and we also have the gonad, the primordial gonad. If there is no Y chromosome, if it's going to be a female, then the gonad develops into an ovary, and then the ovary develops, starts producing estrogen, and then the Müllerian ducts are going to develop into the upper parts of the vagina and the uterus, and then the uterine tubes. If there is a Y chromosome, then the Müllerian ducts will degenerate, and the Wolffian ducts will proliferate. The Wolffian ducts will become the epididymis, plus the vas deferens, and also the seminal vesicle. And then when it comes to the external genitalia, we have the genital tubercle, the urethral folds and grooves, and then the labioscrotal swellings. If it is female, then the genital tubercle will develop into the clitoris. The clitoris is the only erectile tissue in the female reproductive system, and it does become engorged and erect during intercourse. The urethral folds, right here and over here, the urethral folds form part of the labia minora, which are the folds of skin on the inside, and they form as a hood around the clitoris. And then we also have the openings to the vagina and the urethra. And then we have the labioscrotal swellings, which are going to be outside here, and the labioscrotal swellings will form the labia majora. And then for the male reproductive system, whenever there is a Y chromosome, then the genital tubercle forms the penis. Right here, the glans penis. The glans penis is the head of the penis, of the corpus spongiosum, and it surrounds the urethral opening. So the glans penis comes from the genital tubercle. And then the urethral folds, they form the shaft of the penis, which you can see right here. And then the labioscrotal swellings, it forms another part of the shaft of the penis as well as the scrotum. All right, let's follow along on this chart. So if there's a Y chromosome that has the SRY gene, the SRY gene was going to take that bipotential gonad and start producing SRY protein. SRY protein, which then differentiates the gonad into the testes, which are going to have these cells. It develops the interstitial cells and the Sertoli cells. Interstitial cells are the cells that produce testosterone. And then after testosterone is produced, that is what's going to develop the Wolffian ducts. And then we have the Sertoli cells that are going to secrete AMH, anti-Müllerian hormone, which will then cause the Müllerian ducts to regress or degenerate and break down. All right, so the next section is about basic patterns of reproduction. We're going to talk about a little bit about gametogenesis, the formation of the gametes. We have egg cells. The egg cells are the largest of the human cells. They're also called oocytes or ova. These large cells are non-motile. That means they do not move on their own, so they have to be moved. What can happen is that the uterine tube, well, first of all, the egg comes from the ovary. It's ovulated from the ovary. And then the fimbriae of the uterine tube create some sort of circulation of the interstitial fluid, and they collect the egg cell into the uterine tube. And then the uterine tube is lined with ciliated simple columnar epithelium. And the cilia on the apical surface of the columnar epithelium produces a wave-like pattern or wave-like motion in order to create a wave of interstitial fluid to propel and move the egg cell, whether it's fertilized or not, down into the uterus. Now the sperm cells, they are the smallest of the cells, and they are motile or they are able to move on their own. They use a flagellum to swim. And the flagellum, if you remember from anatomy, it's the only human cell that has a flagella, and it moves around like a whip, and it propels the sperm cell forward. Now, the flagella is not movable or motile until after it's been ejaculated, and it's been activated, which takes up to 30 minutes after ejaculation. So gametogenesis. Gametogenesis is the formation of gametes. Genesis means the formation of gametes, the gametes. So how do we make the sperm cells and how do we make the egg cells? Well, it starts in utero, which means it starts even before you were born. The primordial testes and the primordial egg cell, or the ovaries, are producing the egg cells, and the ovaries, the seminiferous tubules, were developing the spermatogonia, which are going to become the sperm cells eventually. And we're going to break it down to male gametogenesis and then the female gametogenesis as well. But they both go through mitosis first. Mitosis is the cellular division. we go through the phases: prophase, metaphase, anaphase, telophase. You can remember that from biology. And what you get at the end of mitosis are two identical cells. They are called daughter cells. So when these cells divide, the spermatogonia and the oogonia, they have 46 chromosomes to begin with. And then when they divide by mitosis, the two daughter cells that are identical also have 46 chromosomes. But after they go through mitosis, eventually they go through meiosis, or some people pronounce it meiosis, either way. But meiosis has two sets of divisions. They both go through prophase, metaphase, anaphase, and telophase. And then they go through the whole thing again: prophase 2, metaphase 2, anaphase 2, telophase 2. And when they get at the end of meiosis, they end up with 4 haploid cells. Haploid means that they have the half of the number of the normal chromosomes. So instead of having 46 chromosomes, they have 23 chromosomes. For male gametogenesis, the sperm cells that are going to become, the primordial sperm cells, the stem cells for sperm are called spermatogonia. Spermatogonia develop and multiply by mitosis. It starts before birth, but then at birth, it stops. So at birth, the spermatogonia stop dividing, and quiescent means it is quiet until they reach puberty. Once they reach puberty and the testes start forming testosterone, then the spermatogonia cells start developing. They start dividing again by mitosis, and then some of them start dividing by meiosis. The ones that are developing by meiosis, they're the ones that are become the sperm cells. So once the male reaches puberty, they start forming sperm cells, and then it stops with death. They don't stop producing sperm cells until the end of their life. For female gametogenesis, it is the formation of the egg cell. They start with the stem cells that are going to become egg cells, which are called oogonia. So oogonia are the primordial cells that eventually will become egg cells. So once the bipotential gonad becomes the ovary, then the cells start forming the oogonia cells. By the time they are 5 months along in utero, this is before birth, they have made the maximum number of oogonia cells they're going to have. So by the 5th month of in utero of formation, they're going to have about 2 million egg cells or oogonia cells per ovary. At birth, a lot of these half of them will have already have degenerated, and so by birth they only have 1 million egg cells. These cells are going to stop in the first stage of meiosis. They are frozen in that step until puberty, and then once they reach puberty, they start continuing with the meiosis. Whenever they reach puberty, the number of oogonia cells are going to be reduced by another half, so they have about 500,000 oogonia cells at that point at puberty. And then at each time during the menstrual cycle, they're going to ovulate 1 egg cell. Now, once they start the menstrual cycle, the first couple of menstrual cycles will not have an egg cell. It's just the process of building up the uterus, preparing for implantation, and then sloughing the lining off the uterus and preparing it for the next month. So after the first couple of months, then they start ovulating the first egg. So that starts at puberty, but it stops with menopause. Once the egg cells have been reduced to around 500 egg cells, then that's the signal for the start of menopause. And with menopause, whenever it starts, the menstrual cycle starts to cease. We'll go over some of this towards the end when we start talking about the menstrual cycle and the uterine cycle. But each month, a whole group of egg cells start to mature and develop into egg cells. One of them will become mature faster. It's kind of like a race. One of them will reach the finish line. The other egg cells that start developing at the same time will degenerate, and they will not become the primary egg cell that will be ovulated. So the primary oocytes, once they go through the meiosis process, the first stage of meiosis, they're going to have one primary oocyte, and then we have something called a polar body. A polar body is just a way for this egg cell to get rid of half of their chromosomes. This polar body is just going to disintegrate. It's not going to be used. It cannot be fertilized. And then they have ovulation. That is the secondary oocyte that will be ovulated into the pelvic cavity, and then the fimbriae will collect it and pull it into the ampulla in the infundibulum of the uterine tube. And then if there's been fertilization, that's whenever the egg cell will continue on with meiosis after the sperm cell fertilizes it, so that we end up with a zygote with 46 chromosomes. Some important things to note with gametogenesis with the male and the female reproductive system. We start with the germ cells, which are either spermatogonia or oogonia. These cells have 46 chromosomes. These are diploid cells, and they're going to divide by mitosis. Let's look at the males. Whenever males start dividing by mitosis, they're going to develop into other spermatogonia cells, again with two daughter cells, the same number of chromosomes, 46 chromosomes. But then eventually the spermatogonia are going to develop into either spermatogonia A or spermatogonia B. If they stay spermatogonia A, then they are going to continue to develop more spermatogonia cells. The spermatogonia B cells start to undergo meiosis. Once they undergo meiosis and they start dividing, at the end product from one spermatogonia cell at the end of meiosis is going to be 4 spermatids. 2 spermatids are going to have the X chromosome, and then the other 2 sperm cells will have the Y chromosome. Now in females, the oogonia cells, they also have 46 chromosomes. They're diploid, just like the rest of the cells. Once these cells start going through meiosis at the very end, after they go through both stages of meiosis, we're going to end up with one main egg cell. The rest of the cells are going to be what they call known as polar bodies, which are not viable cells. See, what the female reproductive system is doing is producing one cell that is the most viable and the most likely to be fertilized. So it's going to be the largest of the cells. It's going to have most of the cytoplasm. It's going to have most of the organelles. The rest that are left over are the polar bodies, which are going to just be degenerated eventually. So once this egg cell that has been developed through meiosis joins with the sperm cell, it forms a fertilized egg cell, which is the zygote with 46 chromosomes. All right, so let's talk about what the brain has to do with reproduction. Well, first of all, both sexes produces hormones that are androgens and hormones that are estrogen. Androgens are going to be the male hormones. Estrogen mainly are going to be the female hormones. So androgens that are dominant in males, they're going to become like testosterone, which comes from the testes. And then there is an enzyme called aromatase. It converts androgens to estrogens. So males produce estrogen as well. It just so happens that they produce testosterone more, which is going to be dominant over the estrogen hormone in the male system. The females, the main hormone is going to be the estrogen. The ovaries produce estrogen, plus the ovaries also produce progesterone. Progesterone is the hormone that builds up the uterus each month in order to get ready for pregnancy, if there is some. And then also the adrenal glands produce some steroids and androgens. So I want to point out that females, when they reach menopause and the ovaries are no longer producing estrogen, then females are producing more male hormones than female hormones, which is why your body starts to change a little bit after menopause, like growing a beard. So the hormone that comes from the hypothalamus is called GnRH, gonadotropin-releasing hormones. We talked about these when we went over the chapter on the endocrine system, way back in chapter 7. So GnRH, gonadotropin-releasing hormone, the origin is the hypothalamus, and it targets the anterior pituitary. It stimulates the anterior pituitary to produce the gonadotropins. That's why it's called gonadotropin-releasing hormone. They stimulate the release of gonadotropins, which come from the anterior pituitary. The first gonadotropin we'll talk about is FSH, known as follicle-stimulating hormone. So, GnRH targets the anterior pituitary and stimulates the production of FSH. FSH then targets the gonad. That's why they're called gonadotropins. FSH will stimulate the seminiferous tubules and stimulate the production of sperm cells. Follicle-stimulating hormone also targets the ovarian cortex and starts to stimulate the developing follicles on the ovarian cortex. And then we have LH, luteinizing hormone. Luteinizing hormone comes from the anterior pituitary and it targets the gonads. It targets the testes and it targets the ovaries. Luteinizing hormone is going to stimulate the production of testosterone from the interstitial cells in between the seminiferous tubules of the scrotum. Luteinizing hormone will also stimulate the follicles of the ovarian cortex to produce estrogen. So luteinizing hormone increases the amounts of testosterone or estrogen produced, whether it's male or it's female. The only other hormone I'm going to talk about is called inhibin. Inhibin comes from either the ovarian cortex, if it's female, or the sustenacular cells, if it is male. But inhibin, which comes from the gonads, will target the anterior pituitary, and it decreases FSH secretion. So it actually will slow down sperm production or slow down ovarian cell development. All right, so we have gonadotropin-releasing hormone that comes from the anterior pituitary, targets the anterior pituitary right here, stimulates the production of FSH and LH. LH is the hormone that targets the endocrine cells in order to produce estrogen or testosterone. FSH is going to target the ovaries and the testes in order to produce the gametes, which are going to be the sperm cells or the sex cells. Now, LH is also used in the developing egg cells. LH is going to create or cause ovulation. LH spikes during ovulation. Okay, so let's review some of the male reproductive organs, and we'll do the female reproductive organs as well. So the external genitalia for males include the penis. That is the copulatory organ. The penis will become erect, and then it'll deposit the sperm cells inside of the female reproductive tract. There is a tube that goes through the penis called the urethra. The urethra is a tube that starts at the bladder, it goes through the prostate, and then it goes through the membranous pelvic floor muscles, then it goes through the penis. Remember, there are three parts to the urethra. There's the prostatic urethra, there's the membranous urethra, or the intermediate part of the urethra, and then there's also the spongy urethra, also known as the penile urethra. The penis is made of erectile tissues, and there's two kinds. There's corpus spongiosum or corpus cavernosum. Corpus cavernosum is on the dorsal surface of the penis, and there's two cylinders of the corpus cavernosum. Most of the penis erectile tissue is corpus cavernosum. Then you have corpus spongiosum. Corpus spongiosum surrounds the urethra. It's on the ventral surface of the penis and actually forms the glans penis. So the glans penis is the head of the penis. It is made of corpus spongiosum tissue. And then there's a piece of tissue that surrounds the glans penis and is known as the prepuce or the foreskin. And this is the part that is removed during a circumcision, not the penis like some people might think. Anyway, and then we have the scrotum. The scrotum is the other part of the external genitalia. The scrotum is like a pouch. It's a fleshy pouch, and it holds the testes. There's a wall, there's a septum that separates the testes, so the right and left testicle are not right next to each other. One testicle actually hangs a little lower than the other, and that is so they don't bang up against each other as you walk. Now, the reason why the testes are hanging inside of the scrotum, outside of the body, is because the testes need to stay at a lower temperature than body temperature. Body temperature stays around 98.6. The optimal temperature to make the most sperm cells is 3 degrees cooler, around 96 degrees. So the testes are kept cooler. Now, if the testes get too hot, there are muscles in place that will relax and allow the testicles to drop further away from the body cavity. And then in temperatures that are real cold, these same muscles will contract and lift the testicles closer to the body cavity in order to warm them up closer to 96 degrees. Now, like we said earlier, the testes will descend into the scrotum, but failure to descend is called cryptorchidism. Cryptorchidism. And that is whenever the testes fail to lower into the scrotum, and if that happens, they can do surgery to correct it, but you don't want to wait too long to do that, because if they stay in the body cavity too long, it is too hot and too warm, and then the spermatogonia cells will die. So the accessory glands to the male reproductive system, we have the prostate gland, which is right below the bladder. The urethra goes through the prostate gland. There's also a duct inside the prostate called the ejaculatory duct. The ejaculatory duct is where all of the secretions are going to come together to form semen. We're going to have secretions from the testes that are going to be the sperm cells. There's secretions from the prostate called prostatic fluid. Plus, there's also a gland called seminal vesicle. The seminal vesicle produces a fluid called seminal fluid, which is also emptied into the ejaculatory duct. So the prostate gland produces the prostatic fluid. Seminal vesicles produces seminal fluid. Most of the fluid of semen, which is the ejaculate, is going to be seminal vesicle fluid. 60% of it is. And then another 30% of it is going to come from the prostate gland, and then a very small percentage is going to be the sperm cells. So the prostatic fluid is full of prostaglandins, and it creates smooth muscle contraction, especially during ejaculation, and then also the smooth muscle contraction of the uterus and the vagina after it's been deposited into the female reproductive system. The seminal vesicles produces fluid. Seminal fluid has fructose in it to help nourish the sperm cells, but it also has prostaglandins in it. There's also clotting enzymes, and then there are declotting enzymes in the prostatic fluid and seminal fluid. The clotting enzymes will make the semen clot right after ejaculation, so that after it's been deposited into the female reproductive system, it clots and doesn't just fall out as easy as you would expect. It sticks to the inside walls of the female reproductive system. And then after about 30 minutes, there are enzymes that are going to break up the clots, and now the sperm cells will be activated, and they'll use the flagella, and they'll find their way through the uterus, through the uterine tubes, and find their way to the to the egg cell. The last male accessory gland is the bulbal urethral gland, also known as Cowper's glands. These glands are found in the floor of the pelvic muscles. They empty into the urethra. The secretions from the bulbal urethral glands are alkaline secretions, so they help lubricate the lumen of the urethra. Plus, they also will buffer any acidic urine still left in the urethra. And also, the bulbal urethral gland will lubricate the tip of the penis to help with insertion into the female reproductive system. So the spermatogonia cells are found inside of the testes, inside of tubules called the seminiferous tubules. So the seminiferous tubules are highly coiled tubules inside each lobe of the testes, and that inside there is where we have the spermatogonia, which will develop into sperm cells. In between, outside of the seminiferous tubules, are interstitial cells. The interstitial cells are the cells that secrete testosterone. So it's the interstitial cells that LH targets to stimulate the production of testosterone. The Sertoli cells are also known as the sustanacular cells, also known as sustanocytes. They are the cells that are going to support the sperm cells. They provide a barrier from your immune system. They provide a barrier called the blood-testis barrier. Inside your blood are white blood cells. White blood cells are designed to destroy any cell that they don't recognize as your cells. And one of those cells would have to be the gametes, because they only have half the number of normal chromosomes. So your own white blood cells will destroy your own sperm cells, so we have the blood-testis barrier to keep the white blood cells away from your sperm cells. And then there's also a protein produced by the Sertoli cells known as androgen-binding protein. Testosterone has to bind to the androgen-binding protein in order to stimulate spermatogenesis, the formation of sperm cells. Now, the seminiferous tubules will eventually lead to the epididymis. The epididymis is on the posterior surface of the testes, and that's where the sperm cells go to mature before they are going to be ejaculated, or if they're not going to be ejaculated, then they'll be degenerated, and then certain parts can be recycled, and other parts will just be broken down. After the epididymis, the next duct is the vas deferens, also known as the ductus deferens. The vas deferens is going to travel up through the spermatic cord, through the inguinal canal, around the pubic bone, around the bladder, and into the ejaculatory duct. So the vas deferens is the tube that they're going to cut during a vasectomy whenever the male wants to become sterile. They'll insert scissors behind the scrotum and just clip off the vas deferens, tie them off, kind of like a tube tying when it comes to the fallopian tubes of the female reproductive system. But anyway, the vas deferens is just a transport tube from the epididymis to the ejaculatory duct. These sperm cells will not move from the epididymis to the ejaculatory duct unless they're provoked to do so during intercourse. And that process is called emission. Emission is the process of gathering all the secretions for semen, and that only happens during the pre-intercourse. Whenever the spermatogonia cells undergo spermatogenesis to form the sperm cells, they form spermatids. So one spermatogonia cell produces four spermatids. A spermatid looks a little bit like this. I'm going to try to draw a picture of it. It's not going to be that accurate. It's going to look like mainly like almost any other cell. That is a spermatid. But eventually the spermatid is going to change and undergo some changes and produce a sperm cell, which will look like that. So to go from this to that is called spermiogenesis. What they're going to do is eject most of the cytoplasm. It's going to put the nucleus inside of the head of the sperm cell. And then there's a sac of enzymes in the outside called the acrosome. So the acrosome is a sac of enzymes on the very head of the sperm cell. And these acrosomes are going to burst upon contact with the egg cell. The egg cell, after it's been ovulated, there's a whole barrier around it called the corona radiata. So the sperm cell has to get through the corona radiata just to get to the outer cell membrane of the egg cell. So that's what the acrosome is for. It'll break down the corona radiata. So the hormones that are required for spermatogenesis, we have the gonadotropins, FSH, and then we also need testosterone. Testosterone is going to be the male hormone, and it's required for spermatogenesis. So semen is a mixture of the secretions from the prostate, from the seminal gland, a seminal vesicle, plus also from the bulbourethral gland, and that all produces semen. 99% of the volume of semen is going to be just the fluid from the prostate and the seminiferous tubules. Only 1% of the volume of semen is actually going to be the sperm cells. But still, per ejaculate, we're going to have over 1 million sperm cells. All right, so here we can see this is the bladder right here. This is the prostate. This is the urethra. The urethra goes through the prostate, so that's the prostatic urethra. And here's the urethra that goes through the pelvic floor muscle, so this is the membranous urethra. And right here is the penile, spongy urethra, and it goes all the way through to the outside. So here are the erectile tissues of the penis. This is the corpus spongiosum. Here's the corpus cavernosum. And then here are the bulbourethral glands, Cowper's glands, that are producing secretions that are going to be emptied into the urethra. Whenever you look at the cross-section through the penis, and again, I cannot help but think this looks like Kenny from South Park, we can see the corpus cavernosum, the erectile tissues on the dorsal surface of the penis. So most of the penis is corpus cavernosum. And then here's corpus spongiosum, which is the spongy erectile tissue that surrounds the urethra. All right, so here are the testes. Inside the testes, the seminiferous tubules. The sperm cells are produced in here, then they're transported to the epididymis, where they stay to mature. After they leave the epididymis, if this is right before intercourse, they call this, if you want to call it the foreplay, the gathering of the fluids. So that once that happens, the epididymis is stimulated to produce an interstitial fluid or a testicular fluid, and then it will then push that testicular fluid through the vas deferens, right here, the ductus deferens, and then deposits it into the ejaculatory duct. Here we have fluid from the seminal vesicle being dumped into the ejaculatory duct right here. So here's the Cowper's gland producing a secretion into the lumen of the urethra. So if we're going to follow the path of the ejaculate, the semen, it's going to go through this way. It's going to go through the membranous urethra, and then through the spongy penile urethra, and then to the outside. Of course, this penis is in a flaccid state, so it wouldn't go just straight down. It'd actually go up and out. Anyway, that's another story altogether. This picture is showing the lumen of the seminiferous tubule. Right here. Here they're showing the seminiferous tubules. Inside of the seminiferous tubules, these are spermatogonia cells. The spermatogonia cells are located closest to the outer surface of the seminiferous tubule. These cells produce or reproduce by mitosis. These are the ones that become spermatogonia A or spermatogonia B. The ones that are A stay on the surface. Spermatogonia B move up, because the spermatogonia B will divide by meiosis until they form spermatids. And then the spermatids go through a whole process called spermiogenesis to form the sperm cells, which are going to then be released into the lumen of the seminiferous tubule and then transported to the epididymis. You see that there are cells in between the seminiferous tubules. These are interstitial cells of Leydig. These are the cells that are producing testosterone. So here's the sperm cell. This is the head of the sperm. Here's the midpiece, and here's the tail. The head of the sperm is where we have the nucleus, where we have the 23 chromosomes. This is the acrosome, which is going to be the sac of enzymes on the very end of the head of the sperm cell. Here's the midpiece. The midpiece is full of mitochondria, and the mitochondria produce all kinds of ATP in order for the sperm flagellum to move around and propel the sperm cell towards the egg cell. So on this left-hand side, we have the composition of semen. The components, then we have sperm. Those are the gametes. They come from the seminiferous tubules. It's a really easy read. There's going to be some mucus, which is the lubricant, comes from the bulbourethral gland, so on and so forth. There's a lot of water, and we have some buffers. We have nutrients. We have fructose coming from seminal fluid and prostatic fluid. The fructose is to nourish the sperm cells. And we have vitamin C. They also help to nourish the sperm cells. There have been studies that show that a lack of vitamin D leads to lower than normal sperm counts. So one of the things that they do for males experiencing infertility and low sperm counts is to increase their vitamin C intake. And then there's going to be enzymes to help clot the semen and then liquefy the semen after about 30 minutes. And then zinc. Zinc is unknown reason why it's found there, but it might provide nourishment for the sperm cell. But they've also shown that men with low zinc counts also have low sperm counts. So one of the other things, other than to increase their vitamin C, is to also increase their zinc intake. All right, some of the secondary sex characteristics in males because of the testosterone. It changes the body shape. They become more muscular. There's more tone to the muscles. So here's we have muscular development. They are able to grow thicker beard and they have more body hair than females. The voice becomes lower on most of them. Mine didn't seem to go through that. But anyway, a lowering of the voice. And then also, it increases the libido. And for those who don't know, libido means sex drive. All right, enough about males. Let's do female reproductive system. We'll start with the external genitalia. First, we have the vulva, also known as pudendum, and that includes two folds of tissue. There's the labia majora and the labia minora. The labia majora is going to be larger. It's on the outside. It covers everything. It covers all the openings to the female reproductive system, including the urethral opening and the vaginal opening. On the inside of that is another fold of tissue known as the labia minora. Labia minora forms a cloak or a hood around the only erectile tissue of females, which is the clitoris. The clitoris is surrounded by all kinds of nerve tissue, and it's right in front of the pubic bone. It gives a pleasurable feeling whenever the two pubic bones from the males and the females mash together during intercourse. But the labia minora is more vascular than the labia majora, so the labia minora has a more pink color to it than the labia majora, which has a more flesh color to it. And then we have the urethral opening, which is between the clitoris and the vaginal opening. That's all considered part of the external genitalia. So internally, we have the vagina. Vagina is the organ that receives the erect penis. So the vagina is the copulatory organ for females during reproduction. There is a folded tissue called the hymen that covers the opening that is destroyed or broken down during the first time with intercourse. The vagina also has a canal for the birth canal. The vagina leads to the cervix. The cervix is the opening into the uterus. The cervix is where they're going to collect tissues or cells for a pap smear and see if these cells are normal or atypical. Atypical cells normally indicate some sort of disease process or cancer that's occurring with the uterus. And then we have the uterus. The uterus is a pear-shaped organ. It's anteverted, which means it folds over towards the front. It is an upside-down pear-shaped organ, and that's where that's the organ that houses the fetus. So that's where the zygote will implant to form the placenta. The tissues of the uterus include the perimetrium, which is on the outside. We have the myometrium, which is the smooth muscle. Then we have the endometrium, which is on the inside, lines the lumen. It's the endometrium that builds up and is sloughed off during each menstrual cycle. It builds up to prepare for implantation. If there is no implantation, then it's sloughed off and we begin the whole cycle again the following month. So there's two parts of the endometrium, if you remember from anatomy. There's the stratum functionalis and stratum basalis. The stratum basalis stays all the time in the endometrium. The stratum functionalis builds up each month and then is left off each month. If it's built up and implantation did occur, then part of the stratum functionalis will form the placenta. Then we have two tubes that lead to the ovaries. They're known as the fallopian tubes. They're also known as the oviducts, and they're also known as the uterine tubes, either way. But they're also known as the fallopian tubes. They lead towards the ovaries. They don't actually connect to the ovary. They just, the opening to the fallopian tube is called the ampulla in the infundibulum. The infundibulum has finger-like processes known as fimbriae that brush over the surface of the ovary. So the fallopian tubes will collect the egg cells, and that is where fertilization will take place. And then here's that fimbriae that I was talking about that is going to, those are the finger-like processes that brush over the surface of the ovary. All right, inside the female reproductive system, here's the ovary. So this is the gonad, the primary sex organs for females. These are the ovaries where we have the cortex that will have the egg cells, plus it also has the follicular cells that surround the egg cells. The follicular cells are the cells that produce the hormones, estrogen and progesterone. These are the oviducts, the fallopian tubes, or the uterine tubes. The opening into the oviduct has these finger-like processes known as fimbriae. You see the fimbriae do brush over the surface of the ovary. Once the egg cell has been ovulated from the ovarian cortex, it is collected by the fimbriae, and it travels through the infundibulum and the ampulla, and then the isthmus, which is the thinnest part of the uterus, a uterine tube, into the uterus, and implants into the upper part of the uterine wall. So here's the uterus. This is the fundus of the uterus, the dome-like part on the top. Then we have the body of the uterus, and then we have the cervix. The cervix is the opening. There's an external os and an internal os. The internal os opens into the uterus. External os opens out of the uterus. Here's the cervix right here. Endometrium of the cervix is this line in the inside. The thickest tissue is going to be the myometrium, which is the muscle, smooth muscle. Then we have the perimetrium on the outside. And then here we have the vagina and the vaginal canal, which leads up to the cervix. All right, looking through a sagittal section, here's the clitoris, which is the erectile tissue. This is the labia minora on the inside and labia majora on the outside here. They cover the openings. They have covers the urethral opening, and here's the vaginal opening right here. So this is the bladder and the urethra. Here's the uterus, the pear-shaped organ, and it folds over on the front side towards the front, so this is anteverted position. This is the cervix, and here's the vaginal canal and the vagina. Here's the ovary, and here's the fimbriae of the uterine tube that brushes over the surface of the ovary, and then here's the fallopian tube, also known as the oviduct, also known as the uterine tube. Whenever we start looking at the developing follicles, the developing follicles start as a primordial follicle. These are the stem cells that eventually become the egg cell and the follicular cells. FSH targets these primordial follicles, and they start to develop into primary follicles. Once they start developing into primary follicle, they are destined to either become the egg cell or to degenerate. The group of egg cells that are being developed are called a cohort. So they start as a race. The primordial follicles turn into a primary follicle, and then as they develop, the follicular cells around them will start to multiply until there's a thick layer of follicular cells that surround the egg cell. Once they develop into a secondary follicle, anything that is not as big as this is going to degenerate. There's usually only going to be one secondary follicle that forms out of the cohort of primordial follicles that all develop together. Eventually, the secondary follicle starts getting bigger and starts developing a cavity called an antrum, which is going to be a fluid-filled cavity. These follicular cells that surround the egg cell, these are the cells that produce estrogen and progesterone. Progesterone targets the uterus, and it builds up the lining of the uterus to become nice and thick and lush. All right, so we keep developing. Eventually, we get a dominant follicle, which has a gigantic antrum filled with fluid. And then we have the egg cell. It pushes the egg cell towards the end or the surface of the ovarian cortex. Once it gets big enough, LH surges, and it pushes the egg cell out. Some of the follicular cells stay around the egg cell that forms a zone called the corona radiata. Now, the follicular cells that are left behind, they're not done yet. These cells that are left behind form what is known as the corpus luteum. And they're shown the corpus luteum as a bright yellow structure because it is full of lipids. The corpus luteum produces copious amounts of progesterone. It forms a lot of progesterone, and it makes the lining even thicker, the endometrial lining of the uterus even thicker and more glandular. So it's full of glycogen and other secretions that help with the egg cell. If the egg is fertilized and implants in the wall of the endometrium, a placenta is formed. The placenta forms a hormone known as HCG, which is a pregnancy hormone. HCG feeds back, and it keeps the corpus luteum alive and well. So during pregnancy, this corpus luteum keeps producing more progesterone. As long as the progesterone levels stay high, the uterine lining stays thick. At the end of pregnancy, the corpus luteum starts to break down. It becomes aged. And once this corpus luteum breaks down, it stops producing so much progesterone. Once progesterone levels start to drop, that means the placenta starts to age and childbirth begins. The uterus will contract and it pushes the fetus towards the cervix and it stretches the cervix. The stretching of the cervix sends a signal to the hypothalamus to produce more oxytocin. So oxytocin is produced in the hypothalamus, but it is released by the posterior pituitary. So oxytocin will then target the uterus and create more contractions. So it keeps stretching the cervix. The more the cervix is stretched during childbirth, the more oxytocin is produced until childbirth. Anyway, let's say pregnancy did not occur. So now the corpus luteum, after a few days, after it's producing progesterone, no pregnancy, no HCG, it starts to degenerate. It becomes a piece of scar tissue known as the corpus albicans. And the corpus albicans produces no progesterone. So once progesterone levels drop during the menstrual cycle, that is when the endometrial lining starts to slough off, and it starts the whole menstrual cycle over again. All right, some of this I've already mentioned on the previous slide, but I'll go ahead and talk about it. The ovary is separated into an outer part and an inner part. The outer part of the ovary is called the cortex. The ovarian cortex contains the follicles, and the follicles surround the egg cells. The medulla, the inner part of the ovary, is where we have blood vessels and nerves. Now let's talk about the follicles. The follicles of the ovary start as primordial follicles, which is basically going to be an egg cell with one layer of follicular cells that surround the egg cell. And they start to get bigger, and they turn into primary follicles. Primary follicles lead to secondary follicles. They keep getting larger. The follicular cells keep multiplying. The egg cell stays the same. It's maturing a little bit, but we're not multiplying the egg cells, just the follicular cells that surround it. And then eventually, we have one tertiary follicle, which is the main cell, the egg cell with the follicles around it. It starts to form the antrum, which is the fluid-filled cavity. The fluid keeps building and building until it squishes the egg cell to the very end. And then we have ovulation, which is releasing the egg into the pelvic cavity. Now, the fimbriae should create a circulation of interstitial fluid to collect the egg cell, but if that doesn't happen, then the egg cell will drop to the floor of the pelvis and then eventually just degenerate. This picture just showing the same thing we went over, but if you're more a visual person, primordial follicles, and it tells you the difference or what the common characteristics of a primordial follicle turns into a primary follicle, again, characteristics, and the primary follicle turns into a secondary follicle, characteristics, and then the secondary follicle becomes a tertiary follicle, which becomes the dominant follicle, and then we have ovulation. After ovulation, the cells left behind become the corpus luteum, and then if there's no pregnancy, eventually it becomes the corpus albicans. Let's talk about the menstrual cycle. The menstrual cycle happens each month. It is a process that happens that builds up the endometrial lining of the uterus and then sloughs it off if there's been no pregnancy. But the menstrual cycle will include what happens if there's been implantation. The menstrual cycle is separated into an ovarian cycle and a uterine cycle. The ovarian cycle is everything that's happening with the ovaries during the menstrual cycle. The uterine cycle is everything that's happening in the uterus during the menstrual cycle. The ovarian cycle is separated into three parts. There's the follicular phase, because the follicles of the cells are dictating what's happening during the ovarian cycle. Then we have ovulation, and then we have the luteal phase. The luteal phase has to do with what's happening with the corpus luteum. And with the uterine cycle, there's three parts. There's menses, that is the discharge of the blood, which is going to be the endometrial lining that's being sloughed off. Then there's the proliferative phase, which is the phase that happens in the uterus to build up the inner lining. And then the secretory phase, that's whenever the corpus luteum is producing copious amounts of progesterone so that we increase the layers as well as create secretions and a lot of glands in the endometrium. We're going to highlight some parts of the menstrual cycle, starting the follicular phase of the ovulation and then the luteal phase, and then we'll look at some pictures to kind of help cement it. But what happens during the earlier follicular phase? FSH is going to be secreted, which leads to the growth of tertiary follicles. So FSH levels start to rise, The FSH targets the ovarian follicles and helps them to grow. These follicular cells start producing estrogen and progesterone. The estrogen, as estrogen levels rise, it's going to block FSH and LH. So as estrogen increases, FSH and LH levels will start to decrease. And this is all to help prevent too many follicles from developing at the same time. Let's see. And then the endometrium of the uterus starts to build up because of this initial elevation of estrogen. And then towards the end of the follicular phase, high estrogen becomes a positive feedback mechanism, which means as the estrogen levels get a lot higher, then LH and FSH starts to surge. Now we're leading to ovulation. And then as the LH and FSH starts to surge, estrogen levels stay high, then the uterus lining becomes really thick, and we're waiting for implantation. At the end of the follicular phase, we have ovulation. Ovulation occurs around day 14. So days 1 through 13 are the follicular phase. Day 14 is ovulation. And then after that, days 15 through 28, that's going to be part of the luteal phase. Basically what happens with ovulation, LH spikes, and then the follicle erupts, the antrum erupts, and it pushes the egg cell out into the pelvic cavity, and where it is going to be collected by the fimbriae of the uterine tube. So after ovulation, we have the luteal phase. So in the earlier to mid-luteal phase, the luteal cells, the corpus luteum, produces inhibin. Inhibin is going to target the anterior pituitary and is going to decrease amounts of LH and FSH. And it produces progesterone, which is going to target the uterine lining to make it thicker, and as well as estrogen will do the same thing. The endometrium is preparing for implantation, so it's going to become really glandular and become really thick, waiting for implantation. If implantation occurs, then the placenta will be formed. All right, in the late luteal phase, when we're leading up to the menses of the uterine phase, if there's no pregnancy, the corpus luteum degenerates, becomes the corpus albicans. Progesterone levels and estrogen levels drop. Then FSH levels and LH levels start to rise. And then the endometrium is sloughed off, which leads to menstruation. That's where there is a discharge, a bloody discharge from the vaginal canal. All right, if we look at this chart, we're looking at the gonadotropin levels, FSH and LH. So at the early follicular phase, FSH levels start to rise, but then they drop. That is because the estrogen levels start to rise right here. And then right before ovulation, FSH spikes, but so does LH. LH spikes way higher. LH is the hormone they're going to use to detect for ovulation detector kits. They'll test your LH levels. So once they spike, then they know ovulation is imminent. So ovulation occurs around day 14, as we see right down there. After that, then LH levels and FSH levels will drop. And then towards the end of the luteal phase, they start to rise back up again because we're going to start the whole thing over again. So let's look at the ovaries, what's happening. So in the follicular phase, we go from tertiary follicle to dominant follicle, and then we have ovulation, and the cells left behind are the corpus luteum cells, which then, when they mature, they start to stop producing progesterone. Let's look at estrogen levels. Estrogen levels will start to rise because LH levels rose, and then it starts a positive feedback mechanism. More estrogen is produced, more and more and more and more and more. So it reaches a peak, and then after ovulation, it drops. It goes up a little bit again during the mid-luteal phase, and then it drops again towards the late luteal phase. Let's look at what's happening in the uterus. There are 3 phases to the uterine cycle. There's menses, then there's the proliferative phase, and then there's this secretory phase. The menses starts with day 1. As soon as there's any type of spotting of blood or discharge of the endometrium through the vaginal lining, that starts the menstrual cycle of day 1. So now we are losing the endometrial lining. The menses phase of the uterine cycle lasts from 1 to 7 days. Some books says 1 to 5 days. It's just a variation. It depends on the female. So during days 1 through 7, we have menses. We're losing that discharge. Then, after that, we're starting to build that lining back up again. So then we have the proliferative phase. The proliferative phase is for days 7 through 14. The proliferative phase ends with ovulation. At the end of the proliferative phase, the corpus luteum takes over. So now we're in the luteal phase of the ovarian cycle. And now it does become thicker, but it becomes more glandular. Produces a nice beta-glycogen, which is a sugar for the developing embryo if there has been implantation. At the end of the secretory phase, once the corpus luteum starts to degenerate, becomes the corpus albicans, then the lining starts to get sloughed off. We start the cycle back over again, day 1 with menses. All right, well, let's talk about sex. Let's talk a little bit about procreation. Let's get some definitions down first. There's erection and ejaculation. Erection is whenever the erectile tissues become engorged with blood. In the penis, it is when the corpus cavernosum and the corpus spongiosum are filled with blood from the penile arteries. And the penile arteries are closed off so that as blood goes in, it can't come back out and it's trapped inside the erectile tissue. In the female reproductive system, it has to do with the clitoris. Now, just so you know that the breast tissue also start to swell during intercourse. Ejaculation is getting the semen to the outside of the body. It's going to be deposited into the female reproductive system or wherever the penis happens to be at the time of ejaculation. Anyway, there are 4 phases of human sexual response. We have excitement, and then there is plateau, and then there is orgasm, and then there is resolution. So let's see what's happening during each time. The term coitus, that is the term for sexual intercourse, also known as copulation. I've used the word copulation in previous classes before somebody says, I don't even know what that means. So copulation, coitus, all means sex or sexual intercourse. Any other words you want to use is up to you. All right, so during excitement, that leads up to the point of intercourse. So during excitement, we have vascular congestion. That is causing the erections, whether it's in the penis or in the mammary tissue or in the clitoris. We have lubrication. We're going to lubricate the tip of the glans penis from the bulbourethral gland to prepare for insertion. There are vestibular glands on either side of the opening to the vaginal canal to lubricate the vaginal canal. And then we have erections as well. That all the excitement. Also, in the body, we have an increase in your heart rate. There's also an increase in your breathing rate. So you're kind of going into a sympathetic nervous system mode, but you're not really going full out sympathetic nervous system mode. You have to be in the parasympathetic nervous system mode in order for erections to occur. All right, at some point after excitement, you're going to hit plateau. During plateau, it's kind of a moment of stillness. The heart rate levels out, the breathing rate levels out. Plateau is a short time right before there is orgasm. In order for orgasm to occur, that is going to create muscular contractions for the ejaculation of the semen to the outside, plus also for females, the vaginal canal will contract as well as the uterus will contract as well. So with orgasm, you have to be in the sympathetic nervous system mode. And as a side note, there are certain antidepressants that suppress the sympathetic nervous system. So if you suppress the sympathetic nervous system, you're unable to achieve orgasm. So there are some people on antidepressants unable to have an orgasm. When it comes to excitement and plateau, and also with orgasm, if you remember the uterus that usually lays over the top of the bladder, so actually over the top of the bladder will actually start to straighten out. The vaginal canal will elongate to prepare for the penis. And then after orgasm, we have resolution. That's where everything returns to normal. The penile arteries stop supplying so much blood. The penile veins are able to open up to drain the blood from the penis. So the penis returns back to the flaccid state. Heart rate returns back to normal. Breathing rate returns back to normal. And then there's no more erectile tissues. For males, there is a time period that's called a latent period for about 20 minutes after orgasm before they can achieve another erection. Now that's just an average. Some might be faster, some might take longer. And then for females, there is no latent period. They could go ahead and have an orgasm right away. Let's take a little look-see at the erection reflex that creates the erection. There are 3 types of stimuli that are going to create an erection. There's going to be tactile stimuli. So there's all kinds of nerve endings that are around the penis as well as around the scrotum and also around the anus, if you want to get technical. The mechanoreceptors will detect stimuli, which then send a signal up to the nervous system, which then creates signals that will go down here to open up the penile arteries to create the erection. So that is a tactile stimuli. But then there's also erotic stimuli, where you see something that is erotic to the individual. It's going to vary on the individual. So just seeing some stimuli will create a signal, which then will send another signal down to the penis to create the erection. Or there's also just thoughts about sex. These thoughts also stimulate those higher brain centers located in the hypothalamus, because your sex drive is located in your hypothalamus, which then sends signals down to the penis to create the erection. So why is this even important? It is because there are certain people that have damage to the spinal cord. There could be a damage in the neck, in the cervical spinal cord, or in the lower back. They could be quadriplegic or paraplegic, either way. However, just because the spinal cord has been severed, they can still create or have erections just by erotic stimuli, watching something, or just thinking about sex. So they're still able to have intercourse. All right, whenever there is an erection, the penile arteries will open, the parasympathetic nervous system is stimulated, the sympathetic nervous system is inhibited. So the sympathetic nervous system has to be inhibited to create the erection. But then once we have orgasm is going to switch this. The parasympathetic nervous system is inhibited, and then the sympathetic nervous system takes over when it comes to orgasm. All right, I put this in here, but there are methods that prevent pregnancy, just so we know these terms, what they mean. Contraception means opposite of conception. So it's something that's going to oppose conception. They're called contraception. Contraception. First, there's abstinence. Abstinence means no sex whatsoever. None. Not just a little, not just the tip, however you want to put it. It is total abstinence prevents pregnancy. That is the only thing that's 100% effective. There is also, they say, abstinence during times of predicted fertility. This is tricky. You have to know your menstrual cycle, and you have to know it really well. And if it was the same every month, exactly the same from puberty until menopause, then you could just go ahead and try to predict your fertile times. But it's not, this is not, definitely not 100% accurate. All right, and then we have sterilization, which includes tying the tubes, tubal ligation. That's where they clip the uterine tubes, tie them off so that the egg cell will not make it to the uterus, or the sperm cells can't make their way to the egg cell. And then we have the vasectomy. That's where they remove part of the vas deferens, which is why it's called the vasectomy. So tubal ligations, female reproductive system. Vasectomy is the male reproductive system. Then there are barrier methods, trying to prevent the eggs and the sperm cells from meeting. The diaphragm is inserted into the vagina prior to intercourse so that the sperm cells cannot get through it and make it to the egg cell. There are sponges that act as a diaphragm. The sponges will be soaked or have spermicidal lubricant inside it, or just using condoms. Condoms fit around the penis so that the ejaculate stays inside of the condom and not be deposited into the female reproductive system. And then there's implantation prevention. That's what an IUD will do. An IUD is inserted into the endometrium, and it keeps it agitated so that there cannot be any type of implantation. So there could be fertilization, but the fertilized egg will not be able to be implanted in the wall of the uterus. And then there's some hormonal treatments like using birth control pills or the birth control injections or using a vaginal ring. All right, so that's the end of this chapter for chapter 26. If you have any questions about the final material, give me an email. Your final will be on Monday, May 4th at any time between 7 and 9 p.m.